All right, well, it's great to be able to speak here. I'll be talking mostly about environmental forecasting. There's a lot of people involved, and I'll mention them as we go. I'll try my best to try to stick to that time.
You would be the first one.
So we all know, if we've been around for the past 2 years, that droplets spread human diseases through sneezing, coughing. And this was long before COVID. So these are little bacteria, and bacteria are very small, but you could fit thousands into a typical size raindrop. A question that I've had working with others in plant disease in the agriculture school is, can droplets spread plant diseases as well? So I'm going to share a little bit from work by others. This is raindrop impact, a wheat disease spread by a fungal spore called Fusarium graminearum. It's kind of a neat, you'll see it again. On these leaves that have the disease, when a raindrop hits and the leaf is wet, you'll get sort of splash. And this is one way that the spores can spread to additional plants. In fact, you might have noticed there was a little vortex that appeared to the right over there. So there's a recent paper in PNAS about that. There are other ways. This is sort of a unique way where droplets can coalesce and then get ejected. And this was some work by Dr. Jonathan Barreco, who's in ME, and David Schmale, who's in SPES, School of Plant and Environmental Sciences. So yes, droplets can spread plant diseases. Plant diseases are spread through either bacteria or fungal spores in a lot of different ways. Many of those mechanisms aren't quite known. One that our group was interested in is, can these plant pathogens aerosolize from aquatic sources? It's sort of a missing mechanism in some of the literature. And we were in particular interested in aerosolization of bacteria from wave breaking. So the movie on the lower right, that's just a small pond here in the New River Valley, and you can see the wind blowing over. And if there were some kind of algal bloom in here, then that could be spreading, and some of these algal blooms have toxins. There's also some thought that bacteria that are related to plant diseases might even be related to the water cycle. So just to say some things about these harmful algal blooms, this is from satellite data from Lake Erie. Lake Erie has had some very large algal blooms. So this is where, due to some influx of nutrients, maybe runoff from agricultural waste, that it just sort of feeds algae to the point that there's an explosion in the population. It could be harmful to humans. It affects the water quality and animals as well. Lake Erie had a very large bloom in 2011, and these are actually increasing in severity and intensity year by year. So we're working with some collaborators, one at the University of Michigan, and then also Dr. Hussein Furatan, who's here in the Civil Environmental Engineering Department, to try to understand how algal blooms form, but then also methods to predict how toxins related to it get spread into the atmosphere and can affect population centers. There's another well-known harmful algal bloom that's called the Florida red tide, because it sometimes appears red when it's in high enough numbers. It's caused by a particular bacterium called Carenia brevis. And when there's wave action, dead bacteria can get airborne, and it causes an instant irritation effect. I don't know if any of you have experienced it. I actually went and was part of a field experiment on the Florida Gulf Coast, and this was a mild day, but I had an N95 mask. This was in 2019. So people were sort of suspicious. Why did I have like a whole stockpile of N95 masks? And this is getting worse and worse. And I'll return to this Florida red tide at the end. Just something about that idea of these bacteria that can participate in the water cycle. There are several. One of them is called Pseudomonas syringae. It's a plant pathogen, but it's also found in snow. It's been found in clouds. It's been found in rivers and groundwater. So it seems to be in every part of the water cycle. Plus, it has an ice nucleating protein, which means that it can, in some sense, cause rain to happen when the conditions wouldn't otherwise be favorable to that. So it might be sort of directing its own transport in some bizarre way. So it's a plant pathogen that can create rain. So just to show you some schematic, these plant pathogens get airborne and aerosolized. They can get into clouds. The clouds can cause rain either nearby or elsewhere, usually nearby. And it is related to the types of crops that are around. So this brings up the tantalizing possibility that you can actually control the microclimate by which crops you grow. I'm going to talk mostly about this project that was by a former PhD student that I co-advised who was in biology. She's now working for the FDA, Dr. Renee Peach. And one of her projects was to describe the droplet production from the laboratory simulated aquatic system with applications to this particular bacterium, Pseudomonas cereum. And so our hypotheses were the number and the size, velocity, and ejection angle of droplets from a simulated wave action environment is going to increase with wind speed. Relative amounts of droplets will also vary, and also the capacity to transport bacteria. So wave breaking at the air-water interface can release droplets by a variety of different mechanisms. The ones that we saw the most were actually bubble bursting and fragmentation. Droplet formation by bubble bursting, if you have a wave that sort of traps some air, then around the bubble will burst, and this will shoot out jet drops as well as some film drops. Also fragmentation, this is where there's ligaments of fluid that will break off and droplets will form. So we had an experimental setup. This was in the basement of Norris for a while, not very big, but we had a flume design and a vacuum that pulled the air across the water surface, and we could control the wind speed just a few centimeters above the water surface. We used a LED backlight and then a photon camera and recorded at about 6,000 frames per second, just a small window. And I'll show you a video of that. These are actually the two mechanisms we saw, bubble bursting, and then also fragmentation. So you'll notice both of these were present, others were as well. This was mostly an experimental study where we digitized and were able to track droplets, look at droplet size distributions, as well as the ejection velocity. This is the droplet flux. So basically, the number of droplets per meter squared per second, it was increasing quadratically. So the red curve is a quadratic fit, and it gives a critical velocity of around 3 meters per second for the wind, below which we didn't have any droplets being produced. We could also get the production mass flux. We binned it by droplet diameter. We could only go down to about 50 microns. And of course, there's a lot of droplets below 50 microns, but that was the resolution we had. We saw some droplets that were greater than 1,000 microns, so 1 millimeter droplets. The point you could see that the main contribution to the mass flux is from large droplets. We looked at the droplet size distribution at 4 different wind speeds, and they were fit pretty well by a gamma distribution. We also looked at the droplet speed and also the ejection angle. The downwind direction is 0, so we're looking at a distribution of the ejection angles. The idea here was we were looking forward to possibly using this as a droplet emission source term for transport models. How are things getting out of the water if we happen to know the wave and the wind conditions, say, on Lake Erie? I had mentioned bubble bursting and fragmentation as being 2 different mechanisms we saw. Going back and looking at it by hand, Renee actually went and found the 2 distributions. We saw mostly for the small speeds, we were able to see these 2 existing simultaneously. But I'm showing in blue the diameter, speed, and ejection angle for bubble bursting and fragmentation. And you see that they're different. The weird thing about fragmentation is that the ejection angle is often pointing down, because the way that fragmentation occurs, things could actually be going back down. So that's not going to really contribute to anything that gets airborne. Bubble bursting, though, seems to contribute more to things that get shot up almost vertically. If they're small enough and wind conditions are right, these things can stay aloft for a while and spread. We were also interested in the bacterial cell flux, so there were some separate experiments in another modified flume where we actually counted the bacterial cell flux. So we put this bacteria in the water, did similar wind conditions, and just looked at the cell flux. So this is the potential for bacteria to move across the air-water interface. If you can't see these numbers maybe, but we're getting into up to 10 to the 9 cells per meter squared per second. And this was work with Regina Hanlon and Dr. Henrik Rota at TU Vienna. And one of our motivations was to apply this to harmful algal blooms. This is a particularly bad day on the coast of Florida. You wouldn't want to be anywhere near the water. In fact, I see beachgoers here, and they're probably miserable. You're wishing they'd gone to California. But a lot of people live in this area. Around 3 million are regularly affected by red tide. But we would like to give them some respiratory forecast, right? So you need to know what's in the water, how's it getting out of the water, and you can estimate things and possibly give a forecast, just like a weather forecast, but for this harmful algal bloom, which, like I said, it's really irritating. Like everything burns and you want to cough. And then when you leave the beach, it goes away. If it's a really bad day though, this can go pretty far inland. We heard from residents who lived 20, 30 miles inland. They said it would affect their daily lives. And if they had an idea of when it was coming, just like a weather report, you don't want to go outside when it's raining. You don't want to go outside when it's one of these very high conditions. So crudely, people have come up with a scale of there's none present in the air, low, moderate, high. And we'd like to be able to forecast that. Several collaborators, Tracy Fanaras, a doctor at NOAA, Klaus Moltner, who's in agricultural economics here, Dr. Eric Holt at Clarkson, and a PhD student of mine, Landon Milieu. We looked at this problem. One of the studies was actually an economic study. And I think this could be useful because it tells you how much people are willing to pay. So this is a willingness to pay study. And local residents in there's these 5 counties on the Gulf Coast of Florida with a population of about 3 million. And based on survey data that was done as part of this study, households said that they were willing to pay from $15 to $45 annually to have a forecast. So think of it as possibly an app where you pay some monthly subscription. So even if it was $1 a month from each household, this is up to $40 million a year opportunity. Nobody's doing this. Somebody should do this. And there's been more and more reports of this respiratory irritation. It's not exactly clear why. These things form in the Gulf, and then they just sort of drift to populated areas. And they've been around for hundreds of years. So we have reports going back 500 years that these harmful algal blooms are just something that comes and goes. It leads to large fish kills as well. So it's not just the irritation. When it's really bad, there's stinking fish everywhere, and it's gross. So we'd like to give people the possibility of being able to forecast they can plan their lives. So some future modeling possibilities would be something like a weather map, where right now you could look on, I use dark sky, that's my favorite. You look on dark sky and it'll sort of tell you in Blacksburg what's the rain looking like. We want to do the same things, just like a weather forecast before these toxin levels. And this is just a schematic that we actually used for the economic study, telling people, how much would you be willing to pay for something like this? Even if you had just 24 hours ahead of time, or just a few hours, or you click on one of those boxes and then it tells you what the irritation level would be. And this could apply just as well to lakes. And there's more and more lakes in Virginia. There's Lake Anna has had a harmful algal bloom. And the people that live around there, they wanted to live on a lake and they're paying a lot for that property. They don't want this algal bloom. So we're not really addressing the mitigation yet, but this is more just being able to predict what's there when you should be outside or not. There's also lakes in New York that are experiencing this. And of course, the Great Lakes. So there's a student, Manu Nimala, who's going to be looking at this to come up with the inland airborne transport model. For that, we need the harmful algal bloom cell distribution on the water surface. The main thing that we've now contributed is this droplet emission rate as a function of wind speed and wave height. So Manu's come up with a Lagrangian stochastic model using weather data, and she's already tested it on field studies out at Kentland Farm, which is another great resource here at Virginia Tech. And we mostly care about the concentration at or near the ground because of the animal human health effects. And like I said, more and more lakes, I think Claytor Lake, it's just a matter of time before there's some kind of harmful algal bloom there. It's the end of my talk. If you want to know more, you can follow me on Twitter. Time for
So you were talking about like this algae specifically being from Florida. Has there been any research into the red tides that occur actually in California on the West Coast?
They're not to the level of severity yet, but I think they will be soon.
Yeah, I'm from there.
I think it's similar. I think it might be the same organism.
It's very similar, yeah.
If you like this video, please like and subscribe. Also check out the links and other information in the description.